Hi everyone, this is going to be the first lesson on the topic of cells for students of GCSE biology. In this lesson we're going to be taking a look at the fundamental building block of all living systems, the cell, and we're going to distinguish between the two types of cells that biologists have categorized, prokaryotic cells and eukaryotic cells. So before we go any further, let's take a look at the learning outcomes for this lesson. And if ever you want to find out what the accessible learning outcomes are for any of these lessons in this biology course, you can go to the course cover page, and then you can find either the lesson page in the course navigation and go there, or you can find the learning outcomes for each individual lesson here in the right hand sidebar under the learning outcomes checklist. So for this lesson, we're going to start by defining what we mean by the term cell, and then go on to describe the differences between eukaryotes and prokaryotic cells in terms of their subcellular structures. Now, in eukaryotic cells, we're going to be looking at some organelles in particular, including the nucleus, the chloroplasts, the mitochondria, and so on. And then we're going to be taking a look at bacterial cells and prokaryotic cells in general, in terms of their subcellular structures, the organization of their genetic material, and the differences between subcellular prokaryotic structures such as the ribosomes in prokaryotes and their equivalents in eukaryotes, and see some of those differences. So let's start by looking at the word cell. Now, biology is the science which deals with living systems, and as such, because living systems are the most complex systems that scientists deal with, it's even more important for biologists to develop a form of language which breaks down those systems into their functional components. And so, in keeping with the tradition in science of looking at the smallest functional units of systems and then building up our understanding from those smallest individual units, in biology we break down our understanding of living systems into the smallest possible units in which life can exist. So the cell is defined as the smallest living unit of matter. Now, in your previous education in science, in your earlier years of your education, you may have come across the 7 characteristics of life: movement, respiration, sensitivity to changes in the internal and external environments, growth, reproduction, excretion, and nutrition. Now, some people consider homeostasis, the maintenance of a constant internal environment, another characteristic of living systems. So depending on who you ask, living organisms and living objects in general are defined as being living if they perform those 7 or 8 characteristic functions. And something that doesn't perform all of those characteristic functions is not defined as being a living thing. So organisms such as ourselves, multicellular living things like human beings, they perform all of those they show all of those characteristics, and they are defined as being living for that reason. Now, the smallest piece of matter which performs all of the characteristic functions of matter that I've just listed is defined as the cell. Now, organisms occur in so many varieties. There are so many bewildering numbers of different types of organisms that we've observed, and many more that we haven't discovered and which are being discovered every day. It becomes necessary for biologists to classify organisms, and perhaps you may have come across some of those classification systems already. You may be familiar with the fact that biologists will divide organisms into kingdoms, for example, and then into phyla, and then into even smaller and more specific categories. However, there is a more modern classification system which defines organisms and classifies them into either being prokaryotic organisms or eukaryotic organisms according to the types of cells that they're made up of, and more importantly, the organization of the genetic material in those cells. So, under this 3-domain classification system, we divide all living organisms, all things that perform all of the 7 or 8 characteristics of life, into either prokarya or eukarya. Now, within the prokaryotes and the eukaryotes, there are 3 main domains. There are 2 prokaryotic domains, and there is 1 domain of the eukarya. So, living organisms are defined as being prokaryotes or eukaryotes according to the characteristics of their cells. Prokaryotic cells, as we'll see, have different characteristics to eukaryotic cells, and they have a different way in which the genetic material is organized. So we have these 3 domains. We have the bacteria and the Archaea domains, which are prokaryotic organisms made of prokaryotic type cells, and we have the domain of the Eukarya, which are made up of eukaryotic cells. Now within that domain we have the various kingdoms. We have the kingdoms of the plants and the animals, we have the fungi kingdom, and we have the kingdom Protoctista, the kingdom of protists. Now this is a scheme which, as I say, is the more modern way of classifying organisms, but in biology this scheme is being revised and debated all the time, and so the Protoctista is no longer considered to be a coherent group. But for the purposes of GCSE biology, this is sufficient for what we're going to be discussing. So the domain of the Eukarya is made up of all living things which are classified into these kingdoms. So, as you might expect, these organisms of these different kingdoms are found in all manner of different habitats and display various types of nutrition. Some of them are autotrophs, organisms that obtain their nutrients from the environment and environmental energy, such as by photosynthesis, and some are heterotrophs, animals that, for example, that eat other animals and plants. So most of the members of this domain, Eukarya, including all of the organisms that exist within all the kingdoms of the Eukarya, most of them are multicellular organisms which are made up of multiple cells, although some, like the protists, are unicellular organisms, and an example of that would be the amoeba, which is a single-celled eukaryote, a protoctistan. Now, fungi can be unicellular or they can be multicellular. So an example of a unicellular fungus is the yeast fungus, but many of them can also be multicellular as well. So let's take a look at a generalized animal cell as an example of some of the features that you might find in eukaryotic organisms. So the plasma membrane of animal cells are often folded into these structures called microvilli. Now this is not necessarily a feature of all animal cells. Some animal cells will be specialized and will have microvilli to increase their surface area for absorption of substances across the membrane. So the plasma membrane is a thin sheet of material made up mostly of lipids with some proteins, and its job is to control the movement of substances into and out of the cell, and also to detect changes in the external environment and then pass information about those changes to the internal environment to allow the cell to react to various things going on externally. So we have the plasma membrane, which sometimes, but not always, may be folded into structures like microvilli. Now we also have another important part of cells which is called the cytosol. Now the cytosol is not the same thing as the cytoplasm. The cytoplasm technically is everything inside the cell membrane, including many organelles as well as the gel-like fluid which makes up the cytoplasm. Now the cytoplasm is a watery gel-like liquid, but it actually has many structures embedded within it other than just the organelles. So you also have very thin filaments of proteins and microtubules going all the way through the cytoplasm, and this is called the cytoskeleton and is responsible for intracellular movement of organelles inside the cell. You also have a nucleus, which is the organelle which contains the genetic information. Now the word organelle is used to refer to the structural functional parts of the cell inside the cell. So you have various organelles, various little organs, which is literally what the word organelle translates into, which perform specific functions inside the cell in the same way that the organs in our bodies perform functions, specific functions within our bodies. So the nucleus, the function of the nucleus is to store the genetic information, and it also serves a number of other functions such as the production of ribosomes. You also have a series of flattened connected tubules and sacs called the rough endoplasmic reticulum. Now it's called rough because it is a system of membranes within which you have these ribosomes embedded, and it appears rough under electron microscopes. You also have organelles called the mitochondria. These are the sites of aerobic respiration within cells. These are the microfilaments that make up the cytoskeleton, and these filaments are contractile. They can change their length, and this can cause the movement of organelles inside of cells and can also move other things around inside cells as required, such as lysosomes, for example. You have another organelle called the Golgi body or the Golgi apparatus, and it's a system of these flattened sacs called cisternae. Now, the function of this organelle is to process proteins that have been produced by the ribosomes on the rough endoplasmic reticulum and also the ribosomes which are found floating freely in the cytoplasm. We have another type of endoplasmic reticulum called the smooth endoplasmic reticulum. It's called smooth because unlike the rough ER, it does not have embedded ribosomes in its membranes. Now, the function of this system is to process lipids for their various purposes in the cell. We also have these vesicles. Now, the word vesicle is a general word in biology that refers to a membrane-bound object containing something or other. So lysosomes are vesicles. They are membrane-bound organelles which usually will contain digestive enzymes such as lysozyme, and these are used for digesting things, breaking things down chemically inside the cell. We also have ribosomes which are freely floating in the cytoplasm. Now, ribosomes are the organelles which are responsible for synthesizing proteins according to genetic instructions from the nucleus. Now, again, you can find ribosomes in the cytoplasm floating freely, or you can find them on the rough endoplasmic reticulum as well. Now, you can also find ribosomes on the outer surface of the double membrane around the nucleus, and the rough endoplasmic reticulum is continuous with the outer surface of the nuclear membrane as well. We also have organelles called centrioles. Now, these are organelles which become important during the process of cell division. These are some of the microtubules which are also involved in the cytoskeleton and its function. Let me just go back there for a second. So all of these organelles and many more organelles that we're going to be discussing later on have specific functions that I've referred to already, and we're going to be seeing examples of how they work in this lesson and also in later lessons of this topic and throughout this GCSE biology course. So this is another diagram showing the generalized structure of an animal cell, which is a eukaryotic cell. And as we can see, there are many other features of animal cells in general. There are secretory vesicles. There are, for example, various other features that a eukaryotic cell may have. Now, this is not necessarily an animal cell, but some protists, for example, will have these structures called flagellae. So this is the generalized structure, but specific types of eukaryotic cells may have other structures and may not have certain other structures. So let's take a look at the organization of the genetic material in eukaryotes. Now, the material which carries genetic information is, of course, the DNA, deoxyribose nucleic acid. So this is a very long molecule composed of two polynucleotide strands which are twisted around in a double helix, and it's the sequence of these base pairs which actually encodes genetic information for making proteins. Now, because the DNA is such a large molecule and contains so much information, in order to package it into a structure as small as the nucleus of a cell, it first has to be made more condensed to fit that large molecule inside that nucleus. So the first thing that happens in eukaryotes is that the DNA, this long, thin molecule, is wrapped around a complex of packaging proteins called histones. Now, these are usually found in groups of 8. It's an octamer. And so you have the histone proteins, and the DNA is wound in a coil fashion around these groups of histone proteins to make packaging units called nucleosomes. So we see here that the DNA is being packaged around these histone octamers to form these nucleosomes. But then this this arrangement is then coiled, and then coiled further, and then coiled even further, until we form these structures called chromosomes. So a chromosome is a structure into which the DNA is packaged, and normally you only really see these shapes of chromosomes shortly before cell division begins. Because the purpose of the chromosome is not just to package the DNA in the nucleus and get it all to fit, it's also to distribute the genes into these separate structures so that when the cell divides into two cells, the two daughter cells end up with the correct numbers of genes. Now we're going to be talking about cell division and chromosomes an awful lot more in later lessons of this course. So this is the way in which the DNA is packaged in a eukaryotic cell. And you'll notice that the DNA is a linear molecule and that it's packaged around these packaging proteins into the nucleus. Now, for all of the rest of the cell's life other than cell division, all you see is a kind of dark staining material in the nucleus. You don't see the material in the nucleus condensed into these chromosomes until shortly before cell division begins. So the DNA is linear. It's wound around many protein complexes called histones to form these packaging proteins called packaging units called nucleosomes, which are then coiled and supercoiled further to form the chromosomes shortly before cell division. Now, being a linear molecule, DNA will have ends to it, and at the ends of the DNA molecules you find these protein complexes called telomeres or telomeres. Now, cells that contain these free DNA ends have developed elaborate methods for DNA replication, and these telomeres serve a protective function to prevent the degradation of the DNA from the ends. Now, telomeres have actually been implicated in aging, and so some theories of aging state that every time the cell divides and the DNA is copied, some of these telomeres are broken off the ends of the DNA, until eventually there are no more telomeres and the DNA starts to become degraded with each cell division. And some think that this has something to do with why cells will age. So, mitochondria. Now, mitochondria are the organelles in eukaryotic cells which are responsible for aerobic respiration, for the release of useful energy from glucose using oxygen. Now, in addition to finding DNA in the nucleus of a cell, actually you can find small loops of circular DNA inside the mitochondria themselves. Now, this allows these mitochondria to multiply themselves inside the cell to provide more energy when the cell needs it, and they can multiply independently of the cell replicating itself. So this mitochondrial DNA actually resembles the small loops of circular DNA that you find in bacteria. So mitochondria are able to replicate themselves, to copy themselves independently of the chromosomes in the nucleus of the cell, and they can carry additional genes to those found in the chromosomes. Now, another feature that you find inside these organelles are ribosomes, and these ribosomes, again, they resemble the ribosomes that are found in prokaryotes, organisms like bacteria. So this together with the fact that these organisms or these organelles can contain these circular loops of DNA, again similar to those found in bacteria, this has led to some scientists proposing a theory called the endosymbiont hypothesis, the endosymbiont theory. And this states that at some point early on in evolutionary history, these were once free-living prokaryotic organisms which entered into a symbiotic relationship with the early eukaryotes. The early eukaryotic cells took these inside themselves. They engulfed these early prokaryotes, and rather than digesting them and breaking them down, there was a symbiotic relationship that was struck between the two types of organisms, where the eukaryotic cell, which enclosed these, provided them with shelter from the external conditions, and in return for that, these that were once free-living organisms and became organelles of the eukaryotic cells provided that eukaryotic host cell with useful energy. So another type of organelle found in eukaryotes, in plants for example, is the chloroplast, and this is the site of photosynthesis. Now, like the mitochondria, these are organelles that have a double membrane and also contain circular loops of DNA and ribosomes, which again resemble the smaller ribosomes found in prokaryotic organisms. This chloroplast DNA again resembles the circular loops of DNA that you find in prokaryotic organisms. And again, this has led some scientists to propose that, like mitochondria, these were once free-living organisms that became engulfed by early eukaryotes and entered into a symbiotic relationship. This time, again, the eukaryotic host cell would provide these with shelter from the external conditions, and these would become the organelles within the eukaryotes that provided those eukaryotes with useful energy, this time from photosynthesis. Now, ribosomes are these small, non-membrane bound organelles found in both Prokarya and Eukarya, and the function of these is to manufacture protein molecules according to instructions received from the nucleus. So, eukaryotic ribosomes have a size in Svedberg units of 80s. Now, prokaryotic ribosomes are smaller. They have an overall size of 70s. And this is the same size of ribosome that you find in the chloroplasts and in the mitochondria of eukaryotic cells. Again, this adds support to the idea that chloroplasts and mitochondria were once free-living prokaryotic organisms that got engulfed and taken into the bodies of the cells of early eukaryotes and struck up that symbiotic relationship. So this diagram gives you a sense of the relative sizes of the eukaryotic and prokaryotic ribosomes, and we can also see their dimensions in angstroms, which is a atomic and molecular unit of measurement of size. Now, an angstrom is 10 to the power 10 meters, so that gives you a sense of the size of these things and their relative sizes as well. So plant and animal cells, these are both eukaryotic cells. The plant kingdom and the animal kingdom, these both belong to the domain Eukarya. At the cellular level, though, there are still some key differences between these two different types of eukaryotic cells. So, for example, plant cells typically will have a cell wall made of a structural carbohydrate called cellulose, and eukaryotic or animal cells will not have a cell wall. We also, in plant cells, typically will have a large central membrane-bound organelle containing cell sap, a watery cell sap called the large vacuole. Animal cells do not contain a central large vacuole. Plant cells typically will be the eukaryotic cells that contain chloroplasts, and animal cells do not have chloroplasts because they don't have a photosynthetic function. Now, animal cells will have features such as microvilli, which you don't find in plant cells, and animal cells also have centrioles. So here we can see that there are specific differences between plant cells and animal cells, and this is not something that you typically find. These are not structures that you find in animal cells, and conversely, structures such as microvilli that you find and also centrioles that you find in animal cells are not found in plant cells, even though both of these are eukaryotes. So, as in all eukaryotes, plant cells possess a plasma membrane which retains the cytosol, the watery liquid within which all of the cell organelles float. So that's the distinction between the cytosol and the cytoplasm. The cytosol is simply a gel-like liquid within the boundary of the cell membrane. The cytoplasm includes that watery liquid, the cytosol, and everything floating around in it. Now, underneath the cell membrane, underneath the cell wall, rather, you have the plasma membrane. And the cell wall is made up of a structural polysaccharide, a structural carbohydrate called cellulose. Now, some other types of cells also have cell walls. So, for example, fungal cells will have cell walls, but fungi, even though they are eukaryotes, they have cell walls which are made up of a different type of material than cellulose. Now, in plants, in multicellular plants, the adjacent cell walls are pushed up against each other and will have these plasmodesmata, which are linkages between the cells on either side of those cell walls. And typically, when you have plants, there's also a layer of material which holds together the adjacent cell walls called the middle lamellum. Now, the middle lamella is made up of a glue-like substance, or a series of substances called pectic substances. So we can see several structural differences here between plant and cell walls, plants and animal cells, which I've already been over here. But you do need to be aware of these differences. So try and keep these differences in mind, but also the fact that they have many similarities. So we can see here that both contain a nucleus, both contain rough and smooth endoplasmic reticuli, both of them contain one or more Golgi bodies and mitochondria, and so on. So let's move on to the vacuole now. So in plant cells, you have this large membrane-bound central organelle called the vacuole. Now this is a fluid-filled organelle, and it's made up, the fluid inside is typically called cell sap, which is actually mainly water with a number of dissolved substances in it, including things like sugars, mineral salts, and also some enzymes as well. Now the vacuole is bound by a membrane, and the membrane is called the tonoplast. So the function of the vacuole is partly as a storage region for the cell, for the storage of both water and also some of the dissolved substances, but it also plays a role in maintaining the shape of the cell and also the rigidity of plants. When there's sufficient fluid in the vacuole, the contents of the cell push up against the cell wall, which causes the adjacent cell walls to push against each other, and this provides rigidity for parts of the plant that require it, such as the stem. And this is why when the plant is deprived of water and doesn't have sufficient water in the central vacuole, that pressure between adjacent cells is reduced, and this causes parts of the plant's structure, such as the stem, to wilt. So we mentioned chloroplasts. Obviously these are the sites of photosynthesis, and these are these large oval-shaped organelles which have double membranes. Now we're going to be taking a look at the structures of chloroplasts in more depth in later lessons as well. Now another one of these organelles that you find in plant cells and typically don't find in animal cells is the amyloplasts. Now the amyloplasts are essentially storage areas for large amounts of starch, and starch is a polysaccharide which is made up of many glucose molecules. So much of the glucose produced by photosynthesis is stored long term as starch in these granule-like structures called amyloplasts. So here we can see a slightly enlarged diagram of the chloroplast. It has a double membrane called the envelope. Within this you have a gel-like substance called the stroma, and within the stroma you have a number of other structures. So you have these flattened stacks of membrane-bound structures called thylakoids. Now the thylakoids contain the chlorophyll, which is responsible for absorbing the sunlight energy and transducing that so that it can be used to produce sugars in photosynthesis. So these are the individual thylakoids here, and they're stacked together into these stack structures called grana. So grana is the plural. A single stack of the thylakoids is referred to as a granum, singular. Now the stroma, as I said, also contains these ribosomes which resemble the smaller 70S ribosomes that you find in prokaryotes, as well as the circular loops of DNA that you find in chloroplasts as well. Now, these lipid droplets are also visible under higher magnifications, and these are simply droplets of lipids, which are kind of storage areas for lipids, which are to be used by the chloroplast later on. So many of the sugar molecules formed by photosynthesis are stored as these starch granules, these starch grains, inside the chloroplast. Now these are not quite the same as amyloplasts, but they serve a similar function here. It's for long-term storage of large numbers of glucose molecules as a polysaccharide, as starch. So this micrograph is an image taken under a transmission electron microscope of a single chloroplast, and as you can see here, you can see the chloroplast envelope here, you can see the stroma containing numerous small ribosomes, you can see a single granum highlighted here, and you can see these long thin membrane membrane-bound sacs connecting the separate grani, and these are called lamellae. Now, the word lamellae or lamellum is a general word in biology meaning a thin structure of some description. Now, in this context, these are the thin flattened tubes which project from each of these grana to neighboring grana. Here we can see a starch grain which comes up as a white object under the micrograph here. And you can also see these dark circular lipid droplets. So chlorophyll and other light-absorbing pigments are located within the membranes of the thylakoids. Now, during photosynthesis, the chlorophyll molecules will absorb light, and the electrons within those chlorophyll molecules will then be excited to higher energy levels, and then the energy carried by those electrons can then be used to synthesize glucose from carbon dioxide and water. So the light energy comes in this direction, and this is the reason why the thylakoids are stacked in this way. It's to maximize the absorption of light energy. So we're going to be talking about photosynthesis in more detail in a later lesson of this biology course that deals with bioenergetics. But for now, just remember that the chlorophyll, the green pigment, and many other photosynthetic pigments which are embedded within the membranes of these thylakoids will absorb, the electrons in those pigments will absorb photons, packets of light energy, and become excited to higher energy levels. And then this triggers a series of reactions called the light-dependent reactions, which involve the conversion of the absorbed light energy into the chemical energy stored in the bonds of glucose. So there's another series of chemical reactions which lead to the synthesis of other nutrients in the stroma, and these include things like lipids and also proteins. So the reactions that occur in the stroma are referred to as the light-independent reactions. Here again we have an electron micrograph taken with a transmission electron microscope, and this shows the structure of a single plant cell. So we can clearly see here the vacuole, the large central vacuole, and we can also see a number of chloroplasts here as well. And the cell wall is located around the edges here, where you can just about see there's a gap between this cell and the adjacent cell with the two cell walls in between. These are the chloroplasts down here, which you can see are packed up against the edges of the cell, and this plant cell, as you might expect, has numerous chloroplasts here. Now, you can also see clearly here the nucleus, which contains the genetic material. Now, at this point in this cell cycle, it's not dividing, it's not undergoing cell division, and so you can't see individual chromosomes. What you can see is this dark staining material, which is made up of the packaging proteins with the DNA wound around them. This material is called chromatin. So chromatin is the word used to refer to the DNA which has been packaged around the packaging proteins. Now, the chromatin becomes organized into those X-shaped structures that you saw before called chromosomes prior to cell division. So this is an electron micrograph of a plant cell to illustrate some of those structures, and this next slide here shows some of the generalized structure as a more kind of cartoon-like diagram of a plant cell. So we can see the large central vacuole with its tonoplast membrane around it. Here we can see the nucleus with its double membrane, and this is surrounded by the rough endoplasmic reticulum, which is continuous with the external wall of the nuclear membrane. Now, inside the nuclei of eukaryotic cells, such as plant cells and animal cells, typically you'll find a region of the nucleus where you have a much denser, larger concentration of the chromatin. This is called the nucleolus. Now, it's the nucleolus, this is where ribosomes are typically produced. You can also see some of the other organelles here. We can see the Golgi vesicles coming from the Golgi apparatus here. These vesicles contain proteins that have been either are going towards the Golgi apparatus ready to be modified, or they're coming from the Golgi apparatus ready to be delivered to various parts of the cell where those proteins are required. Here we can see a mitochondrion, and since this is a plant cell, we can also see the chloroplast here. So this is the generalized structure of a plant cell. And here we can see a comparison between the transmission electron micrographs of two typical eukaryotic cells. On the left-hand side here we have the animal cell, on the right-hand side we have the plant cell here. So you may want to pause here and see if you can spot the specific differences that we've referred to before. Now, in any discussion of cells, we have to get a sense of the sizes of the things that we're dealing with here. So the question here is, how small is a red blood cell? We're all familiar with blood, and we should by now be familiar with the fact that the red colouring of the blood is due to the presence of these red blood cells. So how small is a red blood cell? Well, they can be viewed under an optical microscope, and typically cells and subcellular structures are measured in units of millionths of a meter or micrometers. Now the term micrometer, the prefix micro, can be applied to any unit of measure. So a micrometer is a millionth of a meter, a microgram is a millionth of a gram, and so on. Now the prefix micro is abbreviated using the Greek symbol mu, which looks like a U with a kind of tail on the front. So this is the shorthand for saying micrometer, and 1 micrometer is a millionth of a meter, or 1,000th of a millimeter. So a millimeter is a thousandth of a meter, and 1,000th of that would be a micrometer. So this diagram on the left here compares the various cells and subcellular structures in their sizes, and it also compares them to the thickness of a typical human hair. So the typical human hair is about 100 micrometers in diameter, and so we can see that one of the largest animal cells, the ovum, the female egg cell, is about the size of the diameter of a human hair. So ova are just barely visible under the naked eye in a suspension. Now a cheek cell, the kind of cell that you might swab from the inner surface of your cheeks, is just over half that size. If we compare that to a nerve cells typically are very long, but they're very small in their diameter, in their thickness. So here we can see the thickness of the axon of a nerve cell is just under 10 micrometers. We can see that a sperm cell is approximately this size in comparison to the others, and we can compare that to a white blood cell, which is even smaller still. Finally, a red blood cell is only about 6 to 8 micrometers in diameter, so this gives you a sense of how big these are in comparison to the size of human hair. Now, to get a better sense of the scale of some of these things, I would encourage all of you to look up this simulation on, you can find it on search engines if you just type in the scale of the universe 2. It will take you to one of the results will be this URL here, where you can find this simulation. You will need to enable Flash on your browser, and it's a very useful simulation for comparing the relative sizes of things to scale. So we see here that the average human is 1.7 meters tall, and you can click on any of these objects to find more information about it other than just its size. So you may or may not be aware that there is such a thing as a giant earthworm that can be about 3 meters long. So if we're looking at the size of cells, then we need to go obviously to smaller than the meter. So here we can see a common earthworm in comparison is only 4 centimeters in size. Now again, you need to be familiar and conversant with these units and these prefixes denoting either fractions or multiples of a unit. So a centimeter is 10 to the power minus 2 of a meter. It's 1/100th of a meter. And so this is the size of the common earthworm in centimeters. Now we can go smaller than this, and we're down to the size of the average ant, and now we're down to millimeters. Now the prefix milli means 1,000th, so 10 to the power minus 3 of a meter is a millimeter. If we go smaller still, the largest bacterium is approximately 750 micrometers, so this is millionths of a meter. Now because we're dealing with 750 micrometers, we're at a scale of 10 to the power minus 4 meters, but a micrometer is 1 times 10 to the power minus 6 of a meter. It's a millionth of a meter. So if we go smaller still to the size of an amoeba is 350 micrometers. The human egg, about 120 micrometers on average. And then we get to a skin cell. Skin cells around 35 micrometers. Now we get down to the size of a red blood cell, so about 7 micrometers. Now, if we look at the size of a mitochondrion, which is a organelle of eukaryotic cells, now we're down to about 4 micrometers. An E. coli bacterium, about 2 micrometers. The average chromosome, the X chromosome, for example, it's about 4 micrometers. The Y chromosome is smaller. It's about 1.5 micrometers. Now, if we get down to the size of viruses now, we get to nanometers, billionths of a meter. So a nanometer is 10 to the power minus 9 of a meter. And you need to not just be aware of what these prefixes mean in terms of powers of 10 and fractions, you need to be able to convert between them. So if you have any trouble with that and you're enrolled on my course, then by all means ask any questions that you have on the lessons page, either in the live chat or on that page you can send me a private message and I'll happily respond within 24 hours. And many of the exam questions that you'll be coming across, both in this course and in your within your school or your college, and in your actual exams, will require you to be aware of these units and be able to convert between these units. So I would encourage all of you to take a look at this simulation. You can use it for understanding biology as well as physics, because you can have a look at certain physical phenomena here as well. And you can get to the domain of the very small. You can look at molecules, you can look at atoms, you can even look at things smaller than an atom. So you can look at, for example, protons, neutrons, which are measured in femtometers, 10 to the power minus 5 meters. You can even go down to the weird and wonderful world of the quark when we're talking about scales of zeptometers, incredibly small scales, 10 to the power minus 20 meters here for the bottom quark, and even smaller still, right the way down to the quantum foam and the Planck length. Alternatively, you can scale your way out of this and look at the very large as well. So I would encourage you all to take a look at this simulation, because you can find some very interesting facts about either the very small things in the universe or even the larger things in the universe, such as planetary bodies and smaller moons and dwarf planets, up to stars, up to galaxies and superclusters, and even some interesting phenomena such as the Great Attractor, and right the way to the observable size of the universe. So by all means play around with this, it is very interesting. Okay, so let's now have a look at a closer comparison of eukaryotic with prokaryotic cell sizes. So firstly, we already saw that the red blood cell is one of the smaller eukaryotic cells found in humans, about 7 micrometers on average, and this symbol here means diameter. So if we compare that to a medium-sized bacterium such as E. coli, we find that the E. coli cell, the whole cell, is much smaller still, 2 micrometers. Now there are various other, let me just go back one here. There are excuse me, there are various other sizes of bacteria, and they do get much smaller than this. So Streptococcus pneumoniae, which is a Diplococcus type bacterium, is smaller. It's about 1.25 micrometers. So bacteria do range in size, but you get a sense here of just how small they can be. So one of the first differences that you'll notice between prokaryotes and eukaryotes is the difference in size. Prokaryotic cells typically are up to 100 times smaller than eukaryotic cells. So let's take a look at some more features of prokaryotes here. So prokaryotes are the organisms that belong to either the bacterial or the Archaea domain. These are both prokaryotic organisms, but they are a little bit different from each other. So these again will occupy a very diverse range of environments and habitats, and also show a couple of different ways of modes of nutrition. Now, members of these domains tend to obtain their nutrition by absorption from the environment, by photosynthesis, and also by chemosynthesis, which is a type of autotrophy where the organism will produce sugars by making use of inorganic substances from the environment and the energy released by inorganic chemical reactions in the environment. So the vast majority of the members of these domains are unicellular, they're individual cells. So each organism is one cell, but some of these can form colonies of cells. Now, these are not strictly speaking multicellular organisms because a colony of bacteria is not one organism. But these cells sometimes can join together in colonies to form strands of aggregated cells, for example. So reproduction in prokaryotes, because they are unicellular organisms, this happens by asexual means. This does not involve male and female gametes. And basically the cells will divide to produce new cells. Now, this is not the same kind of cell division that you see in eukaryotes, mitosis and meiosis. This is a different kind of cell division called binary fission. Now, all cells of the bacteria and archaea possess typical prokaryotic features. Now, the single biggest difference between prokaryotes and eukaryotes is not their size, it's the fact that prokaryotic cells, whether you're talking about archaea or bacteria, they lack internal membrane-bound organelles. So, they lack a nucleus because it's a membrane-bound organelle, and pretty much all of the other membrane-bound organelles that you find in eukaryotes are missing in prokaryotes. Prokaryotes also typically will have a protective cell wall, but the cell wall in prokaryotes is made of a different material to the cell walls found in eukaryotic cells such as plant cells. The other main difference here is that the chromosome in prokaryotes is not the DNA is not a linear piece of DNA. It's a loop. Prokaryotic cells like bacteria have a single chromosome made up of one continuous loop of DNA. So, I've mentioned that the bacterial cell wall is made of a different kind of material than the cell walls found in plants, and this is a material called murine. Now, cellulose, which is found making up the cell walls of plant cells, eukaryotic cells, is a structural polysaccharide. It's a carbohydrate. Now, murine is not a carbohydrate, it is a peptidoglycan, which is a different kind of material altogether. Now, another difference is that you find between prokaryotes is that the archaea and the bacteria, even though they are both prokaryotes, they have certain differences between each other. So they both have typical prokaryotic features. So, for example, both archaea and bacteria have a single loop of DNA that makes up their single chromosome. They both lack internal membrane-bound organelles, but the archaea are somewhat different and in some ways are closer to being eukaryotes in their structure than the bacteria. So, for example, the archaea differ from both eukaryotes and from bacteria in that their plasma membranes contain lipids which are not glycerol ester lipids like the ones found in bacteria and also in eukaryotes. They are glycerol ether lipids, and this makes them more stable and less susceptible to being disrupted in temperatures and other extreme environments. They also have more complex forms of the enzymes involved in protein synthesis than their prokaryotic cousins, the bacteria. Now, in the archaea, their genes and their method of protein synthesis are actually closer to the ones that you would typically find in eukaryotes than you find in their prokaryotic cousins, the bacteria. So most of the archaea are referred to as extremophiles because they are tolerant of extreme conditions, either extreme temperatures, extreme salt concentrations, extreme pressures, and so on. So there are various types of extremophiles which are categorized according to the kind of extreme conditions that they live in. So we have halophiles, which are organisms that can live under conditions of high salt concentrations that would kill the cells of other organisms. You also have piezo-philes, which can live under extreme pressures, thermophiles, which can live under conditions of high temperature, and acidophiles, which can thrive in acidic conditions. So in the deepest parts of the oceans where there is no sunlight, it's not possible for photosynthesis to form the basis of a food chain, and the producers at those depths, they can't rely on photosynthesis to create their own food. Instead, they rely on chemosynthesis, which is an example of these would be these hydrothermal vents. Now, On these vents, on these cracks in the ocean floor where you have superheated water coming up and forming these stack-like structures which resemble chimneys, the water at these depths is under such high pressure that even at temperatures approaching 400 degrees centigrade or more, it can't boil to produce steam. And yet you find colonies of these Archaea, these prokaryotic organisms, colonizing the surfaces of these structures at these depths. Now again, at these depths, these organisms can't photosynthesize, and so there is no available light energy for them to do photosynthesis and produce sugars. And so these organisms rely on a different kind of autotrophy, self-feeding, which is called chemosynthesis. And this relies on inorganic substances being released in this superheated water. Things like hydrogen sulfide and methane and also other compounds which are being released in the water coming up from the deeper parts of the Earth's crust. Now these organisms, these Archaea, will use those inorganic chemicals and the energy released from inorganic chemical reactions to power the production of organic substances like sugars and proteins and other nutrients that they need. And they can then form the producers of a food chain at these depths. So this is a so-called black smoker, not because there's black smoke coming from it. This appearance of smoke is due to the particles and the dissolved minerals in the water coming up through these structures, again from deep in the Earth's core. Deep in the Earth's crust, rather. So at these temperatures, ordinary bacteria and ordinary eukaryotic cells would simply be killed by the temperature and also by the pressure and the presence of these somewhat corrosive and poisonous chemicals. But these Archaea, these extremophiles, can live there quite happily. So this is the generalized structure of a bacterial cell. And so there are structures here which are present in all bacterial cells, and there are some structures which are only present in some bacterial cells. So let's take a look at those now. So the structures that you always find is a cell wall made of murine, that peptidoglycan. You also have a cell surface membrane, a plasma membrane. You have these food reserve granules, and you have this long continuous circular loop making up the single bacterial chromosome. You also have these smaller ribosomes. Again, these are 70S ribosomes. And of course you have the cytoplasm. Now, in some structures which are present in some bacteria are these pili or fimbriae. So these are used by the bacterium to attach itself to surfaces. You also sometimes have these smaller loops of DNA called plasmids. Now, these can be used to pass genetic information from one bacterium to another. And so these are quite important in understanding how bacterial resistance to antibiotics is spread from one bacterium to another. And this is something that we're going to be referring to in later lessons. Some bacteria, not all bacteria, but some will have this capsule of a slime-like material which encases around the cell wall. Now this gives an added layer of protection against chemicals in the environment and also prevents the bacterium from drying out if the environment becomes dry. Some bacteria will have this kind of tail-like structure called a flagellum. Some of them will have multiple flagellae. Now, the purpose of this structure is that it rotates like a little propeller to propel the bacterium through liquids to allow it to move. So this electron micrograph shows an E. coli bacterium, and you can clearly see the flagellae of this bacterium. So this is the bacterium that causes foodborne illnesses, and there are hundreds of different strains of E. coli. Now, most of these are harmless and can be found in the intestines of most healthy humans and also other animals. This particular strain of E. coli, the 0175 strain, this produces a powerful toxin which can cause severe illnesses and is usually associated with eating undercooked beef. So, to summarize, bacteria are prokaryotic unicellular living things, although they may stick together to form pairs, clusters, and chains. So the general features of bacteria are that they range in size somewhere between 0.5 and 10 micrometers in length, anything from 20 to 100 times smaller than eukaryotic cells. They are unicellular. They reproduce mostly by a form of cell division called binary fission, which again is not quite the same as the cell division that you see in eukaryotes. They possess a circular DNA molecule that lies unprotected without a nuclear envelope in the cytoplasm. They lack membrane-bound internal organelles, and this is what defines them as being prokaryotes. They possess a rigid cell wall made of a multi-layered peptidoglycan structure, peptidoglycan in particular called murine. They possess smaller ribosomes than found in eukaryotes, and they display infoldings of the plasma membrane called mesosomes, and this is where the respiratory reactions of bacteria take place. They sometimes have numerous finger-like projections coming from the cell surfaces called pili or fimbriae, and this allows them to attach themselves to other cells and also to surfaces, and are also involved in the reproductive process called conjugation. The flagellae, when they're present, have a simple kind of tail whip-like structure and are used in propulsion in locomotion. So let's take a closer look at the bacterial DNA and its organization. Now, the genome of bacteria consists mostly of a single circular DNA molecule, and this forms the bulk of a region of the cytoplasm called the nucleoid. Now, again, bacteria don't have internal membrane-bound organelles like nuclei to enclose the genetic material, but this single loop of DNA lies in the cytoplasm. It's not wrapped around packaging proteins like it is in eukaryotes, and it's a circular loop, unlike the linear DNA strands of eukaryotes. Now, without a nucleus, this loop of DNA is coiled up into a region of the cytoplasm called the nucleoid. Now, in addition to the main bacterial chromosome, some bacteria have these smaller loops of DNA called plasmids, and these are able to replicate themselves independently of the bacterial chromosome and can even be passed from one bacterium to another to allow the transmission of characteristics from one bacterium to another one, such as antibiotic resistance. The bacterial ribosomes are smaller than the ones found in eukaryotes, as we said before. So, to summarize the features then, you might want to take a look at this slide. Some of these we've already talked about. Now again, these are the features that make Prokarya different from Eukarya. But some prokaryotic cells, not all, will have some of these additional features as well. Now, we've spoken about the different kinds of cells that can exist in biology and the ways in which we categorize them as either being prokaryotic or eukaryotic. Now, viruses are acellular. They are not made of cells, and strictly speaking, they're not living at all. Now, the reason why they're not considered to be living is simply because they don't perform all 7 or 8 of those characteristics of living things. The only one that they actually do perform is reproduction. They don't respire, they don't move under their own power, they're not sensitive to changes in their environment, they don't grow, they don't excrete waste substances, they don't feed, they don't take in nutrients of their own, and so they essentially only reproduce. And so even though viruses are associated with living organisms and will affect living organisms, they themselves are not made of cells, and they don't actually perform any of the functions characteristic of life other than reproduction. So if viruses are not living, then what exactly are they? Well, a virus particle, or a virion, is considered to be a small infectious agent which kind of hijacks the ordinary functions of cells and uses those functions to reproduce and create more viruses. So a virus typically consists of 2 or 3 parts. You have the genetic material, which can be either DNA or RNA. The genetic material is typically surrounded by a capsid or a protein coat that protects those genes, and in some virions, such as the HIV virus, the capsid is then surrounded by another envelope, which is made up of the cell membrane of one of the cells that have been infected by that virus. So viruses are classified in various ways, and so if we take, for example, the HIV virus, and again we're going to be talking about infectious illnesses a little bit later on in this course, the HIV virus is considered to be a a retrovirus. It operates in a slightly different manner to most viruses, and as you can see here, you have the genetic material enclosed in a protein coat, and around this we have the membrane, an envelope made up of a similar type of membrane than as you would find on the plasma membrane of a cell. Now, the reason why this membrane is important to the life cycle of the virus is that if this virus is inside a human host, for example, it prevents the membrane, because it's made up of a membrane from one of the human cells. It acts as a kind of cloaking mechanism, which prevents the cells of the immune system recognizing the virus underneath as being a foreign infectious agent and then targeting and destroying it. And this is part of the reason why the HIV virus can effectively evade the normal defenses of the immune system. So all viruses are infectious agents. Not all of them necessarily cause diseases, but they can all infect plant, animal, and even bacterial cells. And they can be very resilient outside of the host cells. They can survive for many years if they're dried into a powder, and can then become activated when dissolved in water and placed on living tissue. And they come in all kinds of different shapes and sizes. So here we can see some of the different shapes of various types of viruses. The tobacco mosaic virus infects tobacco plants, the HIV virus infects humans, and this is the shape of a bacteriophage virus, which actually infects bacterial cells. So viral diseases of humans include the common cold, which is actually caused by a whole variety of different viruses, part of the reason there isn't a cure for the common cold, influenza or flu, smallpox, hepatitis, polio, rabies. These are all viral diseases. And some viruses have been implicated in the formation of cancer as well. So how small is a virus? Well, we actually saw that in the animation that we looked at before. So let's zoom back into the small scale. Down to here. Well, not quite that small. All right, so now here we can see the hepatitis B virus. So we're down to the scale of nanometers. We're talking billionths of a meter here. This here is the HIV virus, 90 nanometers, 90 billionths of a meter. So viruses, they do range in size somewhat, but they are much, much smaller than living cells. So small that 1 million average-sized viruses organized into a row would only be about 5 millimeters long. So the diagram below, again drawn to scale, gives you a sense of just how small a virus is in comparison to one of the smaller eukaryotic cells in our bodies, the red blood cell, and even in comparison to some moderately sized or even smaller bacteria here. So here is a tobacco mosaic virus, which is one of the larger viruses, and you can see just how small it is in comparison to these other cells. The influenza virus and the polio virus are smaller still. So viruses are acellular. They're not made of cells, they're non-living, and they consist of genetic material, DNA or RNA, enclosed in an envelope of protein, and sometimes enclosed in a cell membrane-like envelope. Now, unlike other things, they don't do any of those characteristic functions, and they live by hijacking the internal machinery of cells and turning it to producing more viruses. So after as little as 30 minutes, the host cell that has been infected by the virus can burst to release many hundreds of new viruses, which can then go on to infect other cells. Okay, so I see that the students in attendance have asked a number of very interesting questions, so I'm going to go into the Q&A section with them now. For the purposes of this lesson, if you're watching this as a recording, thank you for watching. I'm going to stop the video recording now. If you have any questions, again, you can simply go to the website, log in, and go to this lessons page if you're enrolled on the course and ask your questions there. If you're watching this on YouTube, by all means, type your questions in the comments and I'll happily answer them there as well. Okay, everyone, thanks for watching, and I'll see you in the next one. Take care.